All right. So far we've looked at evolutionary trends. We've looked at how mechanisms of evolution can give way to new species. And we've looked at it at a very small scale, right? Between two populations, there will be a new species emerging. But this has been happening for a very, very long time. So today we're going to look at basically tracking this throughout the history of life and how we can actually identify the trends of evolution as a result. Okay, before we get started with that, first question. Can't see it. You need me to zoom in. Right, there's an increasing trend of infectious bacteria showing resistance to antibiotics. The implication of this resistance in the treatment of bacterial infections is that, can I get any volunteers? 10? What do you think? Yeah. Avi, what do you think? A person's immune system will adapt to overcome antibiotic resistance. It says compared, right? The answer is B, right? So simple bacterial infections may become life-threatening for patients. So it's essentially think of it like this, right? Bacteria were one of the leading causes of death due to the infections and us being unable to treat them. And that was for the longest of time until the invention of antibiotics. We discovered antibiotics, then we mass produced antibiotics to fight against bacteria. But now the problem is that bacteria is able to be resistant towards antibiotics. So we're literally going back to a time where antibiotics have no use. And that means that even a simple bacterial infection could become life-threatening to humans, right? Because these bacteria, new bacteria, are resistant to more and more antibiotics. So it's like we never had antibiotics to begin with because they can just be resistant towards them. All right. So that's why the answer is that simple bacterial infections can be life-threatening. All right. So today's lesson, we're going to look at the changes in species over geological times as evidenced through the fossil records, right? Basically, how have life as we know evolved over many thousands, millions, billions of years even, and how we can actually track that change through the fossil records that we have been able to find. But before we can get into that, we'll be defining what the fossil record is, how fossils are made, and explain the 4 different types of fossils, along with the 2 different types of fossil dating, absolute and relative. Okay, so all of this is basically talking about how we use fossils and how we actually get information out of them, right? So as you can see, your glossary task is huge for this one because you're going to be dealing with a lot of words that you haven't heard or used in a very long time. You guys probably would have learned about sedimentary rock in year 8, but you probably don't remember it, and it's a different context in this example. So we'll look at that. Other than that, some of these words you would recognize, like radioactive isotope and radiocarbon dating, but we'll go into further detail today about that. Right? So when we think about life as the origination of life, it's basically started with the formation of the first prokaryotic cell. Prokaryotic meaning a simple life form. There's no membrane-bound organelles. There is no complex life. It is basically like a simple bacteria that formed. That would have been the first life form that evolved, that basically emerged on Earth. And then from that moment onwards, the mechanisms of evolution started to turn, and we started to evolve more and more different life forms as a result. So prokaryotes are the first life form. Every other life form that emerged beyond that is basically a descendant of that first cell, right? So all life on Earth is originated from that first cell that emerged, and as a result of evolution and all the mechanisms of evolution, it managed to diversify into the many shapes and forms of life as we know today. So we can't even comprehend how much evolution has occurred over that period of time, because this was about 3.5 billion years ago. So over that period, many changes have occurred over in terms of the life that's formed, right? So unfortunately, that's the sad part, right? Since it was that long ago, we are unable to see most of the life that had emerged, right? So they are basically lost to time and won't ever see the light of day again. But what we can actually do is that we can study what's left behind, which are these fossils, right? Fossils are basically the remains of these living things that have been able to be preserved due to some natural processes that we're going to talk about. But these fossils are what allow us to piece together all the different evolutionary history, the Earth's evolutionary history, and actually better understand how evolution has played a part in developing our final product of Homo sapiens right now, right? So we can also use the fossil records to study relatedness between different species as well, to see how evolution actually drove the life to go towards one direction or the other, and actually track the evolutionary history of life as a whole. So we are going to be looking at it in the scale of geological timeline, where it's going to be millions and millions of years. So hope you like history. So before we get into the nitty gritty, we can also basically, based on the information that we have already of all the fossils and all the things that we found, we can basically start to chart out the history of life on Earth, specifically from the formation of the Earth all the way up until now, which was 2 million years ago, the emergence of the genus Homo. If you're not sure what the genus Homo is, basically the genus that covers all the different species with the name Homo, right? So Homo sapiens, Homo neanderthals, Homo erectus, Homo florensis, and so on. That genus only emerged 2 million years ago. And when you compare it to the scale of life on Earth, it's basically a minuscule part of the life that's emerged. So the genus Homo is so small, and you can imagine us as a species, Homo sapiens, we've only been around for 200,000 years. So in terms of all the life that's emerged, we're nothing more than a speck in the history of life itself. Okay, so if we look at the major events in terms of life and evolution, we can start off with the emergence of the first prokaryote, which happened 3.8 billion years ago. And then what happened is, for the longest of time, nothing else happened, right? Only that prokaryotic cell could emerge and reproduce and exist. No other cells were emerging, because for that period, evolution was going through a slow process of accruing change and trying to diversify. Eventually, one change occurred that is going to be possible imperative for all life that emerges as a result, which is that a bacterial cell was able to photosynthesize. Basically, a prokaryotic cell, we are theorizing it's a bacterial cell known as cyanobacteria, was able to photosynthesize for the first time. And as a result, we know what the products of photosynthesis are, right? We get, what do we get as a product of photosynthesis? Can I get Abi? Yeah. What are the others? What's the energy? Okay, so the one that we're looking at right now is oxygen, right? The fact is, when cells that were able to photosynthesize emerged, suddenly there was a widespread of photosynthesis occurring, where it released so much oxygen, it actually changed the composition of the atmosphere. Before this one period, there was no oxygen in the atmosphere. Specifically, there was other gases like carbon dioxide that was taking the bulk of all the atmosphere. But at this point, the atmosphere started to change. It started to release a huge amount of oxygen, and as a result, all the life that emerged from that could actually use that oxygen to conduct cellular respiration. So that happened 2.4 billion years ago, and for 400 million years, there was no other life still. It's still prokaryotes, and then the first eukaryotic life emerged. So when you're thinking about the difference between prokaryotic and eukaryotic, remember that eukaryotic are basically complex cells. They basically are able to have stuff like mitochondria, chloroplasts inside themselves to be used for different purposes, right? Unlike prokaryotes, they do not have these membrane-bound organelles within them, so they are considered to be simple cells, whereas eukaryotes are considered to be complex cells because they have these membrane-bound organelles inside them, like mitochondria and chloroplasts. So this is the first emergence of those cells, and this happened due to an event we are referring to as the endosymbiosis event. Basically, when two cells became into one, and the bigger cell took in the smaller cell, and instead of eating it, it basically adopted it for itself, and then they merged to become one cell. That's what the endosymbiosis theory is talking about. And then for the longest of time, these cells were just going about doing their own thing, and then suddenly emerged multicellular life, right? So eukaryotic cells, they were just existing as single cell organisms, and they were not going to be joining together for another about 1.1 billion years, and then suddenly it emerged multicellular life forms. These are basically life forms that are built up of multiple cells that are working together to support one life form. So basically, all the animals, all the plants, all the insects are considered multicellular because they're built up of multiple cells, right? In fact, you as a person right now, you're built up of trillions of different cells that are working together to carry out the functions that you need to do to survive. And those kind of cells, those kind of life forms, organisms only emerged 900 million years ago. And then from that point onwards, evolution's gears were basically kicked into overdrive, right? Because there was so much variation that could exist between different species, different organisms, that there was a Cambrian explosion that occurred. This is referred to as a period of high diversity of life due to the mechanisms of evolution, diverging different species all around, right? And then more so different things started to emerge, right? Animals on land, and then mammals, flowering plants, and then eventually the whole genus Homo emerged. So that looking at the history of Earth up until multicellular life, these are the major incidents, the major events that have literally paved the way for multicellular life to form, right? The first thing is prokaryotic cells emerging. Without them, no life would have been possible on Earth, right? The second thing is photosynthetic cells emerge. Photosynthetic prokaryotes emerge, and they basically release oxygen into the atmosphere, and this is considered as the great oxidation event. So a little fun fact about the great oxidation event is because of the fact that these cells were changing the atmosphere, any other cell that could not survive in an oxygen rich environment were basically poisoned to death, right? Think of it like this. Imagine if all the oxygen in the atmosphere suddenly changed into another gas, we would basically suffocate, right? In the same way, due to this one event, the cells that could not survive in oxygen, basically these prokaryotic cells that couldn't survive in that oxygen were basically killed off as a result of this one event that was releasing so much oxygen into the atmosphere. But one good thing for us is that it allowed for aerobic respiration to start to happen. And the cells that were basically using that one benefit are the eukaryotic cells. Okay, these were the cells that could actually carry out aerobic respiration, so they could actually use the oxygen in the air to obtain energy and so on this paved the way for multicellular life to emerge. All right. So far, we're just looking at the history. We haven't really gotten into the fossils yet. So just stay with me for a bit longer. So the emergence of multicellular life obviously led to a wide diversification of life on Earth. And this happened specifically at one period where there was a high amount of diversity of life emerging, referred to as the Cambrian explosion, which happened about 535 million years ago. This is basically the widespread explosion of complex species across the Earth's surface, and that's where a lot of the fossil records that we are collecting from until today are coming from. Basically, during this period, there was so much diversification happening between life that there were so many different fossils that we are still picking up today as a result of this one period, this one event. So another thing we've also realized is that there are basically events that have really shaped the trajectory of evolution, such as an extinction event, right? These are basically it's a period of time where there is a high rate of extinction within a region or within the entire planet itself. So if you see this picture here, what you notice is that basically there's a graph here, right? What the graph is showing is a extinction level. Basically, the extinction rate. It shows how high the extinction rate went at a certain period. And these can be caused due to different reasons, right? In fact, you guys are very familiar with one of the major extinction events, which is during the Cretaceous, between the Cretaceous and the Paleogene period. With this, basically, we're talking about the asteroid that destroyed the dinosaurs, right? So this asteroid was considered a major extinction event. And when we track all the major extinction events on Earth, there's about 7 to 8 that has happened that has caused a high rate of extinction of all the life that was present at that time. So these extinction events have basically been tied together with the changeover of different geological areas. Basically, the boundary between one geological area to the next, we can see that an extinction event has occurred. So we have tied them together as to better understand how the extinction event has changed the evolutionary trend of the different species on Earth. All right. So now we get into the part where I've talked about the different how life has diversified into different forms, and this specifically happened during the Cambrian explosion. And how are we still able to understand all that? How are we still able to get information about something that happened 500 million plus years ago? Is using the fossil records. So these are basically records that we have been able to obtain due to the fossils that have been left behind. So what are fossils? They're basically the remains of organisms that lived a long, long time ago that has been preserved under the earth through natural processes, which allow us to order and map the history of life on Earth. So one of the things we can do with fossils is not just see what the life is or see what it looked like. We can also get an information of when they were around using different techniques, and we can also use them as a way to determine the chronological period of when they were around as well. So conditions there for fossils to emerge for fossils to be able to be preserved, there are some important conditions that need to be met. Because not just anyone or anything that dies can immediately become a fossil. There are specific conditions that need to be met for any remains to become fossilized through the process known as fossilization. All right. So let's look at this simple process, right? So essentially, when we're thinking about fossilization, the only way remains of an animal or a plant can become fossilized is basically it needs to be rapidly covered by sediment. So sediment is basically loose soil that can basically cover on top of the remains of a living thing, right? So when that happens, what is the benefit is that oxygen exposure is greatly reduced along with the microorganism exposure. So that basically slows down the rate of decomposition. So remember that certain bacteria, sorry, not bacteria, certain fungi, they basically decompose living things to get energy. And what are the things that they need to decompose is oxygen especially. So by having sediment cover the remains very quickly, they can basically prevent or slow down the rate of decomposition such that more and more sediment can build up on top of that. So when more and more sediment builds on top of it, layer by layer, what happens is that it will become more and more compacted. So think of it like this. The difference between you swimming at 10 meters and you swimming at 100 meters below the ocean, right? When you're swimming at 100 meters below the ocean, you will feel a lot more pressure acting on you and you can feel it on your ears and your body. But when you're on top at 10 meters, it's not as much. Same thing here. With more and more sediment placing on top of the remains, there will be more and more pressure acting on the layer below. And eventually what happens is that a process known as cementation will occur, causing all that loose soil to get compacted and become into one rock known as a sedimentary rock. And within this rock is where the fossilized remains can take many forms, including the permineralized mold, or past fossils. All right. So the process is very simple. Let's just have one more look at it. So with an example in dinosaur, right? Essentially, Essentially, we have a dinosaur that dies in a river. Why is a river important? Is because rivers have a lot of loose soil that are moving around, right? So sedimentation can happen much more easily compared to if something dies on top of on top of ground. There's not going to be a lot of chances for sediment to form on top if you just dive on the above the ground. It can still happen, but it's more likely for it to happen better when it's happening at a river, because right after a living thing dies on the river, there will be sediment that starts to overlap and cover the bones or cover the remains of the dinosaur. And over time, those that sediment will accumulate more and more on top and form different layers as a result. And because it's forming more and more layers on top, there'll be more and more pressure acting on the bottom layer. And eventually what happens is that the compaction, the pressure that is acting on the bottom layer will make the sediment become compacted and form into a rock known as sedimentary rock. And eventually, different geological processes will cause those sedimentary rock to move around, and suddenly the rock erodes, exposing the fossilized body structures that were buried deep underground. So essentially, there are certain regions on Earth where sedimentary rock has risen up, or geological events have caused rocks to shift around that has suddenly moved these rocks from the bottom all the way to the top back again. And as a result, by due to the natural erosion of soil, you can suddenly see these fossils that are remaining. Right? Any questions so far? All right, moving on. All right. So we've looked at how it happens. So what are the conditions that need to be met for this process to naturally occur, right? First of all, it needs to be protected against decomposers or scavengers. Basically, whatever remains are there, it needs to be protected against the fungi, the decomposers that will basically break down all the remains. So it needs to be buried very quickly so that it can be protected against it, right? Yeah, but it is done through the layering of sediment over the remains. Next is the rapid sediment accumulation. So the best places for this to happen, like I said, are the riverbeds, because this is where natural sedimentation occurs at a much faster rate compared to above the ground. So right now, if something died, a living thing died above ground, there is not going to be a lot of chance for sedimentation to occur on top of the remains. Because it's above ground, there's no loose soil coming out and covering the remains. Whereas if you're at a riverbed, soil will constantly move around and start to cover the remains. And eventually, before you even know it, it has gotten a rapid burial naturally happening. All right. And then the third thing is constant cool temperatures, just to reduce decomposition, low oxygen availability, again to lower the activity of decomposition, and also the low light exposure to also lower the rate of decomposition. So basically, all the conditions are to how can the rate of decomposition be slowed down, and also how can it be buried very quickly. Those are the two major things that need to be done, and these are the major ways that it needs to be followed. Yes. Why? So essentially, yes, but that's not really fossilization. The act of fossilization is talking about also minerals replacing the organic material that's left behind. Basically, as a result of them interacting with the soil, minerals will start to replace the space that was taken up by these organic. So let's say bones, those cells don't always stay the same. What happens when they're buried is that they get fossilized, their minerals get replaced with the natural minerals around the soil. But with the Antarctic, with ice, that's much more of a very pristine preservation where organic matter might not even decay because of the cold temperatures, but essentially that would be much more preserved. That would be much better preserved as well. In fact, that's actually the case with certain cases. I think there was a woolly mammoth that was recently discovered not too long ago, perfectly preserved in ice, because that's what ice would do. Ice would slow down all biological processes to a screeching halt because the temperature is so low that no microorganism, nothing, no natural breakdown, a decomposition process can occur, and also no fossilization will occur because there are no minerals that are replacing the organic material. Okay, they are basically just held there and will never decompose or never go away unless we melt the ice, in which case they do. All right. So when we think about the different fossils, there are a lot of different names for the fossils. Some of them are talking about the way that the fossils have formed, and some of them are talking about what characteristics the fossils have, right? So these first three are talking about specific fossils that are being used in relation to dating. For example, actually no, the trace fossil is an example of a fossil that isn't basically the remains of an organism, but it is the something that indicates the presence of an organism. So basically, footprints, nests, stuff that the thing would have left, living thing would have left behind or built itself, that whatever was preserved, we call those trace fossils because we trace the living thing using these fossils. So it is not the organism itself, right? This is basically anything that it's left behind or anything that it has built that has been preserved. Next is your index fossils and transitional fossils. I'll go into detail about those two in a bit. They're basically used for in reference to dating the fossils, how old the fossils are, which we're going to be looking at in a bit. And then there's the three different examples of fossils: the permineralized fossil, the mold fossils, and the cast fossils. I'm going to ask you guys to look this up yourselves because it's not really incredibly important to know this for your BCE biology. I would still ask you guys to basically make sure you know what are the differences between the three of them. But personally, I'm not really putting a lot of effort into those three examples, right? So far we've looked at the evolutionary trend of living things. We've looked at how there's different periods where life has diversified and how the remains of the life that existed back then is what we are studying until today as fossils, right? So now we know that fossils exist. We know that by looking at those fossils, we get a shape, we get an idea of how the shape of the living thing is. Now we want to see what other information we can derive from these fossils. So the one thing we absolutely love to understand when it comes to fossils is how old they are, right? We need to know how old they are so that we can plot it in the scale of evolution and understand where this life would have emerged and how long it would have existed and how it basically impacted whatever came after that. So that's when we have two methods for aging, for determining the age of a fossil. One is relative dating. So this is a form of dating that where we basically date fossils relatively in terms of each other. So the first relative dating method is using the law of fossil succession, which says that fossils that are closer to the surface must be younger than those that are found below them. So remember the process of sedimentation and how fossils occur, right? Basically, sediment forms on top, it presses against the fossils over many years, and eventually it forms into sedimentary rock. So by that basic logic itself, you can tell that whatever fossil that forms on top will be younger compared to those that form at the bottom, because sediment layers form on top of each other over time. So whatever is forming first will be the oldest, and then whatever comes after that will be younger than that. That is what the law of succession of fossils, law of fossil succession is talking about. And you can see that, yeah, sorry. You can see that here as well, right? So basically, if we see a fossil of a crab at the very earliest level of a sedimentary rock, we can know that that was a very recent fossil that formed, right? But if we saw the fossil of a trilobite or a sunfish or a seashell that formed at a much lower level of the sedimentary rock, we know that fossil formed many, many years ago. And basically, how deep it is within the fossil sedimentary rock is what we can use to determine how old it could relatively be. But this method is not foolproof, right? In fact, this is only telling us how old they are relative to each other, right? Basically, it's not giving us an exact year count. It's basically telling us this fossil is older, this fossil is younger than this fossil. This fossil is older than that fossil. That's all the information we're getting here. We're not really getting a actual timeline of when they existed, right? So we basically use this to determine relative dating, right? We are relatively determining how old the fossils are between each other. And we also supplement this form of relative dating with the existing knowledge of other fossils in that strata. And when I say strata, I'm talking about the layer within the sedimentary rock. So this is one strata, this is one strata, that is one strata. Basically, the different layers within the sedimentary rock is what we refer to as the being the strata. So since different stratas are so distinct from each other, scientists have basically assigned them with particular periods of time. Basically, we refer to different stratas as being from different geological time scales. All right. So if you see here, right, this example here, if you see the year, you can see that this strata over here, you can it's the lowest and the highest strata. And the lower down you go, you can see the age of the strata is much different as well. So between strata Y and strata Y. So if we call this strata X and strata Y, you can see that strata X is much younger, 99,999,000 to 50,000 years ago, whereas strata X is going to be much older, being 150,000 to 100,000 years ago. Any questions about this so far? All right. So when studying relative dating, we use a few fossils, and those are the fossils we were talking about here: index fossils and transitional fossils. These two types of fossils become very important when we are trying to relatively date the different fossils, right? So what is it? What does it mean for a fossil to be considered a index fossil? This is basically a reference fossil that scientists can use to basically determine which period that fossils on the same strata have formed. So if fossil X is an index fossil and we find another fossil in the same strata, we can kind of have a idea of when that fossil was alive. And if we see any fossils above the index fossil, we know that those fossils emerged after this fossil was buried. And any fossils that fall below the index fossil, we can actually have an idea of when it would have been around as well. Basically, these fossils allow us to define the relative age of a target fossil. Basically, if found within the same strata as the index fossil, it can be concluded that they coexisted around the same geological period. And fossils lower than the index fossil would indicate older fossil, and higher would indicate otherwise. They are basically almost like bookmarks within the geological timescales that we can use as a reference to determine how old it could possibly be. Because the thing about index fossils is that, for one thing, we basically already know when they existed. We have information about that already. That's why it acts as a bookmark, because once we know when the index fossil existed, we can kind of have a guess of how old the top fossils are, how old the bottom fossils are, and we can even say that the fossils in the same strata would have existed within that same time period as well. So if we already know that this index fossil existed at around 150 to 100,000 year period years ago, we know that the fossil Y, which was found on the same strata, would probably have the same period where it existed. Okay, so for a fossil to be considered an index fossil, there needs to be four conditions, right? One is that it needs to be very physically distinctive. Basically, we need to be able to identify by just seeing that fossil that, okay, this was the index fossil that we've all agreed upon. This is because needs to be easily identified against any other different fossils they see. That's how it can work as an index fossil. Second thing is that it needs to have had a large population. That whatever living thing that we consider to become a index fossil, it needs to make sure that it was very widespread so that it can be found in a consistent manner wherever we go. And it also has to have existed in many geological areas. So imagine I'm trying to research the fossils that I find in Australia versus the fossils I find in the US. If I can't use the same index fossils to try to relatively date them, then it becomes harder for me to relatively date the fossils I have here. So only if these index fossils have actually existed in many different geological areas can I actually use them as index fossils. Okay, if you're getting confused about this, just remember these are basically the equivalent of a bookmark that you can determine how old that strata is and how old the fossils around it are gonna be, right? And last one is that it only live within a known short period of time. So basically, if it lived for a very long time, it will be found on many different stratas, right? Basically, the longer you live, the more stratas you will be found in, because they could have died millions of years ago, and then a few 100,000 years ago, and then so on, and become fossilized. So for fossil to be considered an index fossil, it needs to make sure that it was only around for a specific period of time, and make sure that it's a short period of time so that we can use that fossil as an index fossil. Who's confused by the idea of an index fossil? Right, let's have a kind of an idea of this, right? So remember, with relative dating, you're basically not using any information other than just being able to determine which one is older and younger, right? That's the only information we had at that point. This is where index fossils came in. So index fossils basically are fossils that have been given a determined age. We already know how old they are, and we already know what period they existed in, right? So in the context of relative dating, where we don't know any other information, we rely on the index fossils to give us a basic starting point. So if we know that in this specific index fossil existed from 100 to 150,000 years ago, we know that anything that falls below it is older than that. And we know anything that falls above it is younger than that. So we can have a idea of the relative age of the fossils within those stratums, right? Okay, I hope so. Moving on. Now is your transitional fossils. Okay, so if index fossils are used to basically relatively date the fossils within the strata, transitional fossils are basically used for forming a link between morphologically distinct species. So basically, if you looked at an orangutan, a chimpanzee, a gorilla, and a man, they have very different bone structures and very different shapes and forms, right? But by looking at the transitional fossils, we can basically see how they have changed to become into a different species, a different genus even, right? So these are basically talking about intermediary fossils between different ancestral species and descendant species, and they can be useful to form a link between the morphologically distinct species. So how does that work is that they basically act as the final puzzle between two very distinct species to know how they will evolve over time, right? So let's look at this in the example, right? Which is this Tiktaalik rosea. Okay, so at first there were these fishes, right? And those were the ancestral species, and the descendant species were those that could walk on land. Okay? If we looked at these two different species, we would say that they are very distinct from each other, that there is no link between these species. But that's when we have our transitional fossils that indicate to us, no, there was a link between these morphologically distinct species, where there were species that were trying to emerge from the water into the land, and they were having features that allowed them to do both, right? They could survive in the water and also somewhat survive in land as well. So that is what these transitional fossils do. They basically act as a puzzle that links the evolutionary trend of different of a particular genus over time, right? So how they have morph over time, you can only track that due to the transitional fossils that inform the link between the ancestral one and the descendant one. Alright. So when we look at the transitional fossils, what we'll notice is that they will have characteristics similar to the ancestral species and also the transitional species, sorry, the descendant species, because they are in between both of them and can have both the characteristics as well. Okay, any questions here? So if index fossils are used in relation to relative dating, transitional fossils, their main focus is to make sure that there is a evolutionary, there's a link to show us the evolutionary change of not even genuses, sometimes it could be even bigger than that, right? It could be entire families that have changed over time. We can use these transitional fossils to indicate that, as seen here, right? So between a gibbon and a human, if you didn't see these different animals, your chimpanzees, your orangutans, and your gorillas, you would say that they are basically unrelated because one has ridiculously long hands and huge palms and very different bone structures. So we wouldn't really link them together until we saw the transitional fossils in between them, showing how they were changing over time to become into the homogeneous as we know it today. Are good? Anyone confused? Anyone need further explanation? Clear as a fiddle. Maintain it sounds. Okay. So now we've looked at relative dating. We are going to be looking at a more absolute way to determine the age, right? Before this, we were trying to basically determine the age or determine the range at which it could fall by using a few different ways, right? First is the law of fossil succession. Second is the index fossils. And third is also using transitional fossils. But now we're going to look at another way to date them, which is giving us an absolute dating. Basically, giving us a precise year count of how old these fossils are. And that is using radiometric dating. Basically, this is using the half-life of radioactive isotopes, as the key in determining how old a fossil is. So this form of dating is much more certain and can give a very clear estimate on the age of the fossils in years. And more is actually still used as one of the most common ways of absolutely dating a fossil. And there are different ways we can do that as well. There are different absolute dating methods as well. But radiometric dating is still considered to be the most common method up until today to date the fossils. So the idea behind radiometric dating is that you need to understand this process first. Okay? If I asked you guys what this process is, who can have a good guess? Yeah. Basically, we're looking at the half-life of radioactive materials and how they break down into simpler molecules. Okay, this is going to be a very quick physics lesson. Basically, radioactive material, they will naturally undergo a decay event to become a more stable element, right? So if it started out as uranium, eventually it will decay into thorium. And by decaying, it also releases energy in the form of either alpha, beta, or gamma particles. Okay. So far you don't really need to know the alpha beta particles thing. The thing that we're focusing on is the parent nucleus, the parent uh, element that it was, and what it becomes into after it decays, the daughter nucleus. Okay, it's important that you guys consider these two things: parent nucleus, decay event, and then daughter nucleus. And that's going to be the main idea behind us allowing us to date the radioactive fossils. Okay, so how do we do this? We are basically comparing the ratio of radioactive isotopes inside relatively stable amount found in the atmosphere, right? This might not be that easy to understand. Let me go through the three principles and hopefully it starts to make a bit more sense, right? So basically, like I said just now, right? Radioactive, they want to break down into different elements that are considered to be more stable, right? For example, carbon 14 is a radioisotope and it wants to be broken down. It wants to decay to become into a stable isotope, which is nitrogen 14. Okay. And it actually switches between carbon to nitrogen as a result of this decay process. So the thing about radioisotopes atoms is that they actually can decay at any point, any time. But by looking at it as a whole, we understand that there's an average rate of breakdown for a radioactive isotope, and that is actually a constant and can be modeled using the half-life modeling system, right? We're basically determining how long it will take for half of the radioactive material that you have to decay to become the more stable version of, so the more stable isotope. So half-life basically describes the amount of time before half of the mass, half of the mass of the radioisotope is broken down into a predictable and stable product. Okay. So essentially, let's look at this example here, right? So in this example here, carbon 14 is your radioactive isotope, and it wants to break down, it wants to decay to become nitrogen 14. And the half-life of carbon 14 is known to be 5,730 years. This is basically how long it takes for half of the radioactive isotope atoms to become into nitrogen 14. Okay, that is what half-life means. Okay, and after this period, it will take another 5,730 years for the half, whatever remaining half, to become into nitrogen 14. And when I say remaining, is that if there are 4 atoms of carbon 14 left, it will take 5,730 years for half of them to become into nitrogen 14, and then another 5,730 years for the half of whatever remaining is to become into nitrogen 14, and so on. So let's say you have 500 grams. Actually, no, I'll just show the picture to come after this. So, like I said, right, carbon 14 is a radioisotope that has a half-life of 5,730 years. This means that after 5,730 years, an organism's or after it has died and it's not replenishing its carbon 14 deposits, it will basically start to lose whatever carbon 14 atoms it has, and it will take 5,730 years for half of the carbon 14 in your body to decay to become nitrogen 14. And we can actually use that fact to basically do backwards calculations to determine how many years has it been since the living thing died. Because the thing about the carbon 14 within living things is that as long as you're living, you will keep replenishing the carbon 14 within your body through your natural processes. Your eating, your breathing, all this will replenish your carbon 14 in your body. And that's the same with any living thing as well. But as soon as you die, as soon as you're not doing any of those processes, whatever carbon 14 you'd have had at that point is going to be fixed. And beyond that, at that point, the carbon 14 will start to decay at a fixed rate for 5,730 years to get to its half-life, and then it will redo that step again for another 5,730 years, and again until the carbon 14 level in your body is so low that it can't be accurately used to determine the age anymore. So we can actually kind of have an idea of what it looks like right here. So basically, remember the parent isotope that is the original radioactive isotope that was present. And then the daughter product is basically what it is going to be decaying into, right? So if these red orange dots are your carbon 14, the teal dots are your nitrogen 14, right? So we start off with all the atoms being carbon 14, right? And then once decay starts to go through and one half-life has basically elapsed, half of all the atoms have decayed into daughter products, which are your nitrogen 14s. Okay, and then there's still remaining 1, 2, 3, 4, 5, 6, 7, 8, 10, 12, 13, 15, 16. So there are 16 atoms of carbon 14 left, and it takes a whole other half-life for half of that 16 to become into nitrogen 14. Okay, so what's remaining here? 1, 2, 3, 4, 5, 6, 7, 8. So it will take another half-life for half of that 8 to become into nitrogen 14, and another half-life for half of that 4 to become into 2, and then another half-life for 1, and then yeah, eventually at that point it's no point using it because you can't reverse to determine how many atoms were remaining, how many atoms you started off with, or how you don't have enough information to work backwards. If you're only remaining, you've gotten to a point where it's like one radioactive isotope element, sorry, radioactive atom present. So it matters, it is important to consider what your dating period is when you're determining what type of radioisotope you're going to be using to date something. Okay, so essentially, since this idea is so useful in dating, we have basically determined a plethora of different radioisotopes that can be used for absolute dating, and we determine which one to use based on the dating period of whatever fossil we have. If we know our fossil is not going to be super old, we can use carbon dating, right? Basically, that is dating that has that is dating using carbon 14, which has a half-life of 5,730 years. But if we know our fossil is going to be much millions of years, even hundreds of millions of years, we might be better off using uranium 235 and lead 207 as our radioactive isotopes for dating, right? Because the half-life of this uranium 235 is 700 million years. So it'll take 700 million years for half of the radioactive isotope to become into the daughter nucleus, which is your lead 207. All right. So when would we use that is basically when we want to date uranium containing materials, shells and corals and so on. Basically, they've been around for 100 million years. And okay, I need to pick up a pace. So basically, by determining the ratio, the ratio can be used to work out the time that would have been needed for it to pass down to facilities. And this amount of breakdown, which is the age of the fossil, right? Okay, just one more minute, guys. Let me just get through this very quickly. All right. So the gradual decay of radioisotope is integral to determine the accurate dating of fossils and rocks. By comparing how much of the original radioactive isotope has decayed, we can work backwards to determine how many half-lives have elapsed, and as a result, we can determine how many years is that approximately going to be. All right. Yeah, you guys can read the rest on your own. Relax.